If an average globular protein was of the size of a tennis ball, a typical animal cell would be as large as a stadium. 2. Oblique rays of light are focused on the specimen in dark field microscopy. 3. The light used to excite a fluorescent molecule carries greater energy and has a shorter wavelength compared to the light that is then emitted from the molecule. 4. Sequence-specific staining of nucleic acids can be achieved by in situ hybridization of probes that are attached to fluorescent dyes. However, if general DNA staining is desired, a fluorescent organic molecule such as DAPI can be used. 5. Fluorescent or luminescent ion-sensitive indicators, many of which are small molecules, can be used to measure cellular ionic concentrations in real time. 6. A protein can be genetically engineered as a fluorescent fusion protein, and then imaged in living cells by fluorescence microscopy. 7. Quantum dots are used as an alternative to organic fluorochromes such as Psi3 and Alexa dyes because they are brighter, their emission spectra can be precisely fine-tuned, and they have a longer lifetime and bleach more slowly. 8. Two approaches, confocal microscopy and deconvolution microscopy, have been devised to deal with the problem of blurring in light microscopy that comes with imaging thick specimens. Comparing the two approaches, confocal microscopy is faster, but requires brighter illumination. 9. Consider an engineered chimeric protein made from fusion of three proteins, a blue fluorescent protein, BFP, a calmodulin binding peptide, and a green fluorescent protein, GFP. Calmodulin is an abundant calcium binding protein in eukaryotes. Once bound to calcium ions, it can recognize the calmodulin binding peptide in the fusion protein, change conformation, wrap around the peptide, and bring the BFP and GFP components in close proximity. This results in fluorescence resonance energy transfer, FRET, between BFP and GFP. Accordingly, the fusion protein is a genetically encoded calcium indicator that redshifts its emission wavelength in the presence of calcium. 10. Single molecule detection by fluorescence microscopy is limited by the presence of an excess of out-of-focus fluorescent molecules. The TIRF microscope uniquely overcomes this limitation by exciting only in-focus molecules via an evanescent field. 11. Electron microscopy samples are often chemically fixed before dehydration, resin embedding, and sectioning, but they can also be fixed by rapid freezing, in a way that precludes ice crystal formation, to ensure minimal damage to the original cell structures. This be done by high-pressure cooling, plunging into liquid nitrogen, spraying with a jet of liquid propane, or through contact with a copper block cooled by liquid helium. 12. The presence of carbon in a sample provides the lowest electron density. 13. Scanning electron microscopy generally has a greater depth of field, is usually smaller, cheaper, and simpler, and detects electrons that are scattered or emitted from the surface of a specimen. Transmission electron microscopy is used to create electron microscope tomograms. 14. You have generated strains of Drosophila melanogaster that are expected to show interesting developmental phenotypes such as misplaced organs in the adult fly. However, some of these phenotypes are not readily seen with light microscopy. You therefore fix each mutant fly, dry it, coat it with a thin layer of gold, and place the entire fly into an electron microscope for viewing. You would use an EM microscope instead of a TEM microscope. Study Guide. Chapter 10 Membrane Structure. 1. The typical thickness of a lipid bilayer such as the plasma membrane of our cells is 5 nm. 2. Cholesterol, phosphatidylinosytol, sphingomyelin, and ganglious IgM1 are all normally found in a eukaryotic membrane. 3. Flip-flops are very rare for phospholipids, but cholesterol molecules flip-flop more often. 4. Liposomes do not fuse with one another spontaneously when suspended in an aqueous environment because the hydration shell of the polar head groups of the lipids needs to be removed. 5. With longer hydrocarbon chains, the phase transition temperature increases, that is, the membrane can freeze more readily. In contrast, introduction of double bonds in the fatty acids chains creates kinks in the chains and makes it harder to pack them together. At physiological pH, removal of serine adds a negative charge to the head group, also tending to limit tight packing. The effect of cholesterol is more complicated, but at the high concentrations found in eukaryotic plasma membranes, cholesterol also prevents the hydrocarbon chains from crystallizing. 6. Bacterial plasma membranes are often composed of one main type of phospholipid and lack cholesterol. 7. The two monolayers of the plasma membrane in a human red blood cell have different overall electrical charges. with negatively charged phospholipids, for example phosphatidylserine, normally enriched in the inner monolayer. 8. Many cells store lipids in droplets of varying sizes. These droplets are enclosed by a phospholipid monolayer, instead of a bilayer. 9. Glycolipids such as gangliosides may contain oligosaccharide chains with negatively charged residues, are found to constitute about 10% of the total lipid mass in the plasma membrane of neurons, are found in the extracellular leaflet, facing away from the cytosol, in the cellular membranes, and affect the electrical environment of the membrane. 10. Transmembrane proteins are sometimes covalently attached to a fatty acid chain that inserts into the membrane. 11. Integrins are single-pass integral membrane proteins in the plasma membrane of animal cells and are involved in the interaction of the cell with the surrounding extracellular matrix. The transmembrane part of an integrin molecule folds in a conformation with maximal interchain hydrogen bonding. 12. The Greek letter beta barrel arrangement results in higher structural rigidity compared to Greek letter alpha helices. The Greek letter beta barrel proteins vary in their size and the number of strands, and only some of them form membrane-spanning pores for transport. 13. Bacteria rhodopsin changes conformation in response to light. 14. Although functionally diverse, rhodopsins, bacteria rhodopsin, channel rhodopsin, and G-protein coupled receptors share an overall architecture. A cooperin is an entirely different structure of protein. 15. While examining the crystal structure of a membrane protein, you find several phospholipid molecules bound to the protein. You know that these lipids are thought to help stabilize many membrane proteins, may enhance the crystallization of the bound membrane proteins, interact specifically with the protein, and can have head groups of various sizes and charges depending on the protein. 16. In intestinal epithelial cells, the different plasma membrane domains are separated from each other by special barriers. The asymmetric distribution of membrane proteins resulting from these barriers is functionally important. Study Guide. Chapter 11 Membrane Transport of Small Molecules, and the Electrical Properties of Membranes. 1. The permeability of a protein-free lipid bilayer to various molecules depends on their properties. The following molecules in order of lowest permeability into cells to highest permeability into cells is as follows, from left to right: RNA oligonucleotide, ADP, Na+, glucose, urea, O2. 2. Cells not have membrane transport proteins for O2 because oxygen can dissolve in the lipid bilayer and diffuse in and out rapidly without the need for a transporter. 3. The intracellular concentration of potassium is typically higher than the extracellular concentration. 4. Imagine a small synthetic vesicle made from pure phospholipids enclosing an interior lumen containing 1 mm glucose and 1 mm sodium chloride. If the vesicle is placed in pure water, H2O diffusion into the vesicles would happen faster than glucose or sodium chloride diffusion out. 5. In contrast to transporters, the channel proteins in cellular membranes can only mediate passive transport. 6. A Na+-independent Cl, HCO3 exchanger in the plasma membrane normally functions to lower the pH of the cytosol. 7. Many amino acids in our diet are absorbed via the transcellular transport pathway by the intestinal epithelial cells. This process requires ADP hydrolysis by the Na+K+ pumps in the basal and lateral domains of the plasma membrane. 8. The F-type pumps family of APAs is structurally related to the turbine-like pumps that acidify lysosomes and vesicles. However, they usually function in reverse, generating ADP from ADP and PA using proton gradients across membranes. 9. The ABC transporter family is the largest among membrane transport proteins and includes some channels as well as pumps. The multi-drug resistance protein is a member of ABC transporter family. 10. P-type pumps are phosphorylated at a chiasp residue in each transport cycle. The sodium-potassium pump is a member of P-type pump family. 11. V-type pump are responsible for the acidification of synaptic vesicles. 12. Na+ glucose synporter, Ca2+ pump, Na+K+ pump, and bacteria rhodopsin are all electrogenic transporters. The Na+-independent Cl, HCO3 exchanger is not an electrogenic transporter. 13. Most eukaryotic ABC transporters are involved in exporting small molecules from the cytosol. In this subset of the ABC transporters, the small molecule binds better to the ADP-free transporter. 14. The multi-drug resistance protein is a pump that hydrolyzes 2 ADP molecules per transport cycle. 15. An ion channel always mediates passive transport, is ion selective, is typically several orders of magnitude faster than a transporter, and is usually gated. 16. Changing the membrane potential can be achieved by the transport of a small number of ions across the membrane and does not require concentration changes. The resting membrane potential in most cells is close to the K+ equilibrium potential, and is negative inside, because the plasma membrane is most permeable to this ion. 17. A potassium channel conducts K+ ions several orders of magnitude better than Na+ ions, because the Na+ ion is too small to interact with the channel in a way that facilitates the loss of water from the ion. 18. A cooperant does not pass ions such as Na+ through because the Na+ ions do not favorably interact with the wall of the pore and therefore cannot be dehydrated. 19. A cooperant has a pair of key asparagine residues located on the wall almost halfway through its pore. These residues simultaneously bind to the oxygen atom of a passing water molecule, rendering it unavailable for hydrogen bonding. As a result, the channel cannot conduct protons. 20. MSCS and MSCL are mechanosensitive channels that open in response to membrane tension. 21. A neuron's repetitive firing rate is limited by an absolute refractory period, during which a new action potential cannot be generated. Inactivation of voltage-gated Na+ channels is chiefly responsible for this limit. 22. If the extracellular concentration of sodium ions is artificially decreased for a neuron, the resting potential hardly changes but the action potential peak is lowered. 23. Compromising the inactivation mechanism in voltage-gated Na+ channels results in action potentials that have extended depolarization phases.